Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about the basics of cell signaling. So in previous videos we've established the importance of cells in the body being able to establish communication with each other, whether it be sensors in the skin communicating with the brain, cells in the brain communicating with cells in the heart. Cells in the body need to be able to communicate with each other in order to maintain homeostasis. However, what we haven't really covered is the issue of how do cells actually communicate with one another at the molecular level. So there's going to be a couple of things necessary. First, we need to consider the cell that is doing the signaling, or in other words, is sending the message. The cell that's sending the message needs to both synthesize and secrete a signaling molecule. Yes, the message in this case is going to be a molecule that moves from the cell that is sending the message towards the cell that is going to receive the message. In this case, you can see a cell has synthesized this green molecule here. The synthesis, of course, will happen inside of the signaling cell. The signal will need to exit the cell either by exocytosis or by simple diffusion, depending on exactly what our signaling molecule is. Our signaling molecule can be anything from a short peptide, a fully folded protein, an amine like epinephrine, or a steroid like testosterone. Then, if we consider the cell receiving the signal needs to have the correct receptor protein for that particular signaling molecule. So if you consider this signal right here, this cell is capable of receiving and interpreting the contents of this message because it possesses the correct receptor for that particular signal. So just having a receptor is not sufficient. You have to have the correct receptor. The relationship between signaling molecule and receptor is very important, as we are going to find out in this unit. So there you can see the receptor. So the nature of how the cell that is signaling relays the message to the cell that is receiving the signal is the signaling molecule travels from place to place until it finds the cell with the correct receptor. So this engagement between the receptor and the signal can be anything from a hormone to a neurotransmitter to a variety of other things. We tend to use the catch-all umbrella term ligand when we're talking about a molecule that binds to a receptor in this way. But engagement between the receptor and the ligand will stimulate a change in the target cell's behavior, and this in some way is going to lead to our desired response. Future videos, when we talk about signal transduction, will focus on specifically how does that response get generated, but we can save that for another time. Now, when we talk about different modes of signaling, it's the nature of how the ligand travels from cell to cell in the body that's going to determine what type of signaling pathway we're using. For example, the way in which a neurotransmitter hits its target cells is going to be very different from how a hormone hits its target cells, neural versus endocrine in that regard. I mentioned this before, but the way in which the ligand stimulates the change in cell behavior is called signal transduction. We're not going to cover it in this video, but we will be covering it in the very near future. Now, one last thing I want to clear up here is that in my previous example, I showed you a cell that is a target for a particular signal, and that target cell's receptor was located in the plasma membrane. This is not always going to be the case. It often is, but it's not necessarily always going to be true. In this picture, you are seeing a target cell that has both a membrane-bound receptor here in red and an internal receptor here in purple. This purple receptor is just freely floating around inside the cytoplasm of this target cell. The idea here, though, is that the location of the receptor for a given signal is going to very much depend on the chemical properties of that particular signal or ligand itself. Let's see what we mean by that. First, let's consider a hydrophilic ligand. If you've forgotten what hydrophilic means, this is a ligand that is very polar, very water-soluble, but The consequence of that is that this ligand, because it is hydrophilic, will not be able to cross the plasma membrane. If the goal here is to convey the contents of this message to the inside of the cell, we can't get that to happen by having this ligand move across the membrane. So the only option is for the receptor for this ligand to be embedded in the membrane itself and facing towards the extracellular fluid. So in this way, the receptor can engage the ligand without the ligand having to cross the plasma membrane itself. Now, again, how the message still gets relayed inside the cell, that's an issue for signal transduction, and I promise we will cover that in the very near future. But on the other hand, let's consider a case where our ligand is hydrophobic. This means the ligand is not going to be very soluble in water, but the benefit is that hydrophobic ligands usually can cross the cell membrane by simple diffusion, as you're seeing here. Because the ligand can cross the membrane in this way, it can find its receptor waiting for it in the cytoplasm and then immediately engage with it without having to deal with a membrane-bound receptor. Now, in the future, when we talk about signal transduction pathways, we'll be talking about the types of responses that you can usually expect from a target cell once its ligand engages with the receptor, but we'll go ahead and mention this for right now. When you're dealing with hydrophobic ligands, most of the time we're going to be talking about steroids as our best example of hydrophobic ligands. The response is usually pretty easy to predict, and we've actually talked a little bit about this in a past video. So when the hydrophobic ligand binds to its receptor, the complex of the receptor and the ligand forms an active transcription factor. So once this engagement between ligand and receptor happens, the transcription factor translocates inside the nucleus where it binds to a specific promoter of a specific gene on the DNA, and this is going to lead to transcription and translation in the eventual synthesis of a new protein. So when you're dealing with a hydrophobic ligand binding to its receptor, the response is almost always going to be the target cell makes a new protein that it was not making before, and the change in cell behavior and the change in physiology is because of the function of this new protein that's just shown up. Okay, that's going to do it for this video. If there's a list of vocabulary terms here you probably ought to be aware of. For checking your understanding, number 1, what is the relationship between a ligand and its receptor? Number 2, how do secreted ligands travel to their target cells? Number 3, what makes a target cell capable of responding to a signal or a ligand? And 4, how can we predict the location of a receptor if we know what the signal is? So that'll do it. If you have any questions, go ahead and drop them in the comment section. I'll see you next time. So long.